So our research was on microplastics and nutrient pollution and assessing water quality in Spartanburg. My name is Samantha Omar.
And my name is Lopez.
So when looking at microplastics and nutrient pollution, it is very important to look at the common causes of pollution. Industrial activity and urbanization are two of the leading causes of land and water pollution. In our research, we chose to focus on urbanization and how that has an impact on pollutants found in both land and water. So urbanization involves the construction of roads, buildings, and other similar surfaces that prevent water from soaking into the ground. This leads to increased runoff, which can carry pollutants into bodies of water. Runoff is water from rain or irrigation that flows into these bodies of water. In cities, roads and sidewalks do not absorb that water, so the water will end up going into lakes and rivers and carries the pollutants along with it. So for some background about nitrate and phosphate, their chemical compound structures are displayed at the bottom. Nitrates are a form of nitrogen and phosphates are a form of phosphorus. Nitrate and phosphate are essential nutrients for plant growth in agriculture. Nitrates primarily come from fertilizers and phosphates primarily come from fertilizers and detergents and soaps. Although they both have important impact on agriculture and they help with plant growth, if they are in excessive amounts, they can have significant environmental and health impacts if not managed properly. So among these environmental impacts is a process known as eutrophication. It is when excessive levels of both phosphorus and nitrates are found in the water. These nutrients will stimulate the rapid growth of algae, and that will prevent sunlight from reaching the aquatic organisms found at the bottom of the water. And so when decomposers break down the organic matter, they will release, or they will use up a lot of oxygen from the water, and that will lead to low oxygen levels, and that will lead to detrimental impact for aquatic organisms.
So the other half of our project was microplastics, and so when we talk about microplastics, we're talking about plastics that are less than 5 millimeters in diameter, so pretty small. And these can come from a variety of sources like landfills or litter, and even some of the materials and clothing that we wear. And we don't really have a great way of dealing with them. So through like water treatment processes or anything like that. So they're pretty ubiquitous and they're found pretty much anywhere and everywhere in the environment. So in terms of some of the exposure and effects, so one of the problems with these microplastics is that they stick to other organic pollutants, and they also stick to heavy metals. And so what tends to happen is that you get this effect called bioaccumulation, where a fish eats the microplastic, and then someone eats the fish, and then all of a sudden that person is building up these microplastics in their body. And in one study that we found that was pretty recently done, they found that microplastics were in blood vessels, and these microplastics actually cause an increased risk of heart attacks. So for our study specifically, we had 3 sites. We had the Lower Chinkapin, Glendale, and Upper Chinkapin. So we chose the Lower Chinkapin because it has the highest impervious surface area that we could find. So sitting around 50%. So that means there's a lot of roads nearby, a lot of sidewalks, that sort of thing. And then we chose Glendale because it's more forested, less impervious surface area. And then Upper Chinkapin was supposed to be our like middle ground.
So as Matthew said, our water samples were collected from 3 sites, Glendale, Upper Chinkapin, and Lower Chinkapin. And from these samples, we looked at DO levels, dissolved oxygen, analyzed pH, and nitrate and phosphate levels. Dissolved oxygen refers to the concentration of oxygen found in the water. And as I mentioned before, processes like eutrophication will lead to lower oxygen levels, which will in turn impact all these aquatic organisms. For methods of measurement, phosphate and nitrate levels were measured by an instrument known as a spectrophotometer. So what a spectrophotometer does is it shines the wavelength of light on these nitrate and phosphate levels, and nitrate and phosphate samples will absorb a certain wavelength of light. And the levels of absorbance correlate with how much concentration of nitrate and phosphate are found in these samples. So a higher absorbance will correlate with a higher level of nitrate and phosphate, and a lower absorbance will correlate with a lower level of nitrate and phosphate.
So one of the questions we wanted to answer was how do these sites compare to recommended values in the area? So this bottom row right here shows the recommended values for all of the different things that we tested, and then the highlighted are areas of concern. So we can see that all three sites had high values for phosphate, and then lower Chinkopin, upper Chinkopin had high values for nitrate, and then that pH at the lower Chinkopin was a little bit lower. So the pH is typically an indicator of sort of the health of an ecosystem, and so if you have fish can be more sensitive to pH depending on the species, and so at this sort of range of around 6, you might see some fish struggling to survive in that pH. And then nitrate and phosphate are kind of what we're measuring as sources of pollution. So that seems to indicate that there might be something going on there. And then in terms of the effects of urbanization, so we have Glendale, our less urbanized site, so a lot more impervious, or a lot less impervious surface area, so able to sort of absorb that groundwater more. We see that they have a higher pH than the lower Chinkopin, and then we see for nitrate that the nitrate levels at Glendale are a lot lower than the lower Chinkopin.
And so, as we've seen before, we had high phosphate levels found in 2 of our sites and high nitrate levels found in all 3 of our sites. So looking into that and further implications, that will we can correlate pollution with these high levels. So we must look at ways to decrease pollution levels. And as seen in our results from Glendale and lower Chinkopin, phosphorus was not significantly higher. However, we had significantly higher levels of nitrate and lower levels of pH. And that correlates urbanization because with pollution, because lower Chinkopin is a lot more urbanized than Glendale is. And looking forward at methods, we can look at longitudinal sampling, so looking at these water samples for a longer period of time. So when looking at different water samples, we might have had different weather conditions. Rainfall could correlate with more runoff going into these water samples, and so that could correlate to higher nitrate and phosphate levels that we measured. So if we look at our water samples over a longer period of time, we can get more accurate results.
So back to microplastics, we collected mud to test for the microplastics, and we collected these with the petite ponar or hand trowels. So the petite ponar is that thing right there. And basically what we did was we threw it off the bridge at Glendale, we lowered it into the water, and then it would scoop up the sample from the bottom, and then we would hoist it back up, and that was our sample. And then for the shovel, we just kind of got mud from the riverbank, and then we let them dry in the oven for a week. And then we did what was called density separation and organic degradation. So we created a solution that was a lot denser than a lot of microplastics, and basically all of the microplastics just floated up to the top, and then we filtered off that liquid portion, and we took the filter and we put it in what's called a Fenton reaction, and what that would do is basically degrade all of the organic compounds, so then we were just left with our microplastics. And these are some of our results. So you can see sort of these different fibers here that are pretty dark compared to kind of the rest of the area. And one of the things we learned is that microplastics are actually really hard to detect. We kind of spent a long time over the microscopes trying to figure out oh, is this a microplastic? Is that a microplastic? So there's kind of a lot of gray area because this is still a really, really new kind of area of study. And so we found that there wasn't really super significant differences between Glendale and lower Chinkopin, so not really tied to urbanization as much. But we did have an outlier replicate, which is shown here, with 20 microplastics in a sample. And some of the challenges that we faced was that a lot of these samples are, we're getting 3 40-gram samples, so 120 grams out of all of Glendale. So that's really, really small in comparison. And then plastic identification. So again, I kind of touched on that a little bit earlier, but one of the things that we could do is heat up a needle and put it on the plastic, and if it curls up, then we could say like, oh yes, that's a microplastic. And then throughout the different sediment types, so we have like sand or something that's a little bit finer, like a clay or something like that. Sand was a lot easier to work with and a lot easier to identify different things, whereas those finer particles kind of hindered us from being able to see the microplastics as well. So overall, it's kind of hard to draw a distinct conclusion for the microplastics, but I think in terms of the future, we could look at ways to sort of optimize how we do our sampling and how we sort of analyze these things. So yeah.
Thank you.
Questions for our presenters? Let's go with Dr. Sally. And then Dr. Arrington will come to you in a second.
So was it so dilute that you couldn't get a concentration of microplastics, like how many micrograms per deciliter or something like that?
Do you want to go or you want me to go? Okay, so yeah, the thing is, like, we're looking at it under the microscope, and so I think to have to kind of try and pick out with tweezers like the different microplastics would probably have been more time consuming than it was actually worth. And I think also, like, in terms of, why don't we do like water sampling or something like that? A lot of just the sheer volume of water that we would have to collect would be sort of too much. That it would be a lot to do.
It's too dilute.
Yeah.
Good.
Actually, you have a question?
Yeah, I was going to ask on that, kind of on the same note, is there a standard that exists, like you can purchase a standard solution that has a certain amount of microplastics and you can put that on the slide, look at that and kind of calibrate to know what you're looking at?
So we had talked about having a standard or whatever, but we looked into it, and because it's still relatively new, they were very expensive to buy a standard. We had also talked about kind of shredding up, different types of plastic. We just kind of never got around to it, but yeah, good thoughts.
Other questions from back to back?
So I know nitrate and phosphate runoff is mostly agriculture, but is there anything individual consumers can do on a personal level to help mitigate their contributions to that?
Well, yeah, because as I mentioned before, nitrate comes primarily from fertilizers that we use. Phosphate comes from things like detergent and soaps as well as fertilizers. So we can minimize pollution, which will minimize that runoff that goes into those bodies of water. And we actually have a slide on pollution prevention. So we can minimize these when looking into that.
Yeah, so I think also just kind of being mindful of what you buy and what's in the products that you buy is probably the most important thing. I think there's only so much you can do as an individual, and I think you're probably going to need more substantial policy changes or more substantial decisions in sort of how you build in terms to really get at kind of the root of the problem. So.
All right, wonderful. Let's give them a hand.